here. Have to hit the got it button. I met Jing, I don't know how long ago, and we've been working together, amazing scientist, but she's an infectious disease epidemiologist with expertise in molecular and clinical epidemiology, and she focuses on chronic infection and aging. She's completed her medical training in China, earned a PhD in epi. So I didn't know you were an MD, PhD. So your PhD was in epi from Drexel, and you did your postdoc training in infectious disease in epidemiology here at Hopkins. Dr. Sun is, co-investigator on a number of, some of the largest HIV cohorts in the U.S., including the ALIVE study, as well as the Max Wise combined cohort study. She co-leads the aging working group within MWCS. She supported NIAID, KO, has additional supports from NIA and NEI, and she's really been focusing on exploring the influence of biomarkers such as function, multi and comorbidities in people with HIV. So super excited to hear you speak today. Jing, I will pass it to you.
Great. Thank you so much for the kind introduction. So today I'm going to talk about association of mitochondrial DNA copy number and cognitive impairment among people with and without HIV. So I think this is we're going to do primarily focusing on this one study, but I'd like to also talking a little bit more broader in terms of like means have like mitochondrial dysfunction of people with HIV in general. So okay, so a little bit background. So because of the setting of today's talk, so brain health complication is really persist in people with HIV. Primarily, HIV penetrate the brain during early HIV infection, and as many of the early data show that people with HIV has severe neurocognitive impairment due to the HIV infection. And thanks to the, the effective antiretroviral therapy, this severe neurocognitive impairment has been much less often, much lower prevalence nowadays. However, despite these very effective ART treatment, about 30 to 60% of people, 50% of people with HIV still experience some cognitive impairment, which is more common among people with than that in people without HIV. And in fact, Raha and Leah and teams has demonstrated so many of these leading epidemiological studies showing the risk and then of people with HIV living with, the cognitive impairment among people living with HIV. And then the cognitive impairment has a major impact in terms of like quality of life and aging related and adverse outcome among people aging with HIV, such as, frailty, phenotype, hospitalization, and mortality. So my focus has been studying mitochondrial function and mitochondrial genetic among people with and without HIV. So ever since my postdoc here at Hopkins, I have been really fascinated by this tiny little organelle that living inside of our human body. So a little brief recap, mitochondria contain 37 genes that's separate from the nuclear gene, and then it encoding 13 protein and 2 rRNA and 22 tRNA. And then the remaining 1,500 genes required for mitochondrial function are encoded in the neural genome. So basically there is a tight association between the mitochondrial function and mitochondrial genetic variation with the nuclear genetic variation. And some of my previous study has demonstrated that mitochondrial genomics can influence the mitochondrial function and protein production. And then some of my previous study in the MAX cohort showing that mitochondrial copy number, mitochondrial genetic variations such as haplogroup and haplotype has been associated with like frailty and physical function decline among people with HIV, also showing a significant association with insulin resistant and diabetes incident in max. So, and as we know, mitochondria play a central role in the production of ATP, and it has been known that it regulates multiple bioenergetic process and signaling pathway in across nearly all of the organ system. And here today, we're going to talk about an emergent biomarker, mitochondrial DNA copy number. It is typically considered as a marker of mitochondrial biogenesis. And so basically, the idea is that you're trying to get an average, like how many mitochondrial copy in each cell in your peripheral blood. So the compelling reason for people using this biomarker in a research setting is because it's really readily available, and you just basically need some peripheral blood tissue and trying to get extract their DNA and trying to get a ratio between the mitochondrial DNA copy number to the nuclear DNA. And in the previous study, especially, it shows that mitochondrial DNA copy number may indicate mitochondrial depletion, reduced energy reserve, and oxidative stress. And then in several of the study down in my collaborator Don Arkin's lab, in the ERIC cohort and MESA, shows that mitochondrial copy number is linked to frailty, neurocognitive disease, cardiovascular disease, and mortality in the general population. So why am I interested in mitochondrial copy number or mitochondrial function in general among people with HIV? So some of my previous study in the alive cohort has shown that mitochondrial DNA copy number is reduced among people with HIV. For example, in this paper that we published, we showed that the mitochondrial DNA copy number showing, the y-axis here is the adjusted mitochondrial copy number within the cohort, and the y-axis here is the across different group. The first panel showing in this figure, showing a comparison by the people with and without HIV. So as you can see, people with HIV on the right hand side, the box plot showing a significant reduced mitochondrial DNA copy number compared to the left bar, that's people without HIV. And then we further see that this mitochondrial copy number is further reduced in a dose response fashion based on the HIV disease severity. For example, among those people with lower CD4 cell count, with higher CD4 cell count in this middle bar here, is people with HIV with CD4 cell count greater than 500 cells per UL. And that is a very comparable copy number compared to people without HIV on the very left. But then once you see those people with CD4 cell count 200 to 500 or less than 500, you see the further reduction on mitochondrial DNA copy number across the group. And same thing among when you comparing people with detectable and undetectable HIV viral load, and it really clearly showed that people copy number are reduced most significantly among those people with poorly managed HIV, with detectable viral load, and with a poor immune recovery. And then I think this this graph showing the distribution by their ART use, and I think this is basically a proxy for their HIV viral load or CD4 recovery across the group. And using these longitudinal data, we even further find out that there is a longitudinal trajectory of the mitochondrial DNA copy number over age. And then among those people older than 50 years old, we can see that the blue bar here across are those people with without HIV. And then as you can see that the mitochondrial DNA copy number across the observation window seems like fairly stable. They don't have a significant reduction for the copy number over age. So here we are trying to look at extrapolate the age from 50 to 80. But then when you look at the different group by people with HIV, and you can see a clear trend that mitochondrial DNA copy number are further reduced when people are aging with HIV. So you may ask, why is this relevant? Like, it seems like just a biomarker that's, like part of our sort of general observation. However, like what we see is when you look at mitochondrial DNA copy number and HIV, and looking at some of the outcome here, such as mortality, we see that there is a very clear chance among those people with low mitochondrial DNA copy number and HIV infection, you see a significant decline, higher hazard of all-cause mortality within the alive cohort. And of course, like the solid line here are showing the people with HIV. So the green solid line are the people with high mitochondrial DNA copy number with HIV. The blue solid line is median middle mitochondrial DNA copy number with HIV. And then the red line here, which associated with nearly 60% reduced, like only 40% of survival by the 15 years of observation window, are the people with low copy number with HIV. So this is a very striking differentiation. And as you can see, like even those people without HIV shown in the dotted line in this figure, you see that the mitochondrial copy number seems like in dose response fashion predicting their mortality risk over time. And here on the right panel, I'm showing another paper that we published with one of my students that we're looking at mitochondrial DNA copy number in with one of the aging-related outcomes such as kidney function decline and incidence of chronic kidney disease in the alive cohort. And we have another survival curve showing that people with low mitochondrial DNA copy number is associated with increased incidence of chronic kidney disease in the alive cohort. And additionally, this is the glomerular filtration rate, EGFR figure here. And then on the y-axis, that's the EGFR the value. And then the x-axis is the follow-up time of the study. And the red line showing here is representing the low mitochondrial DNA copy number group. And the blue line here showing the higher mitochondrial DNA copy number group. And as you can see that the trajectory of the EGFR decline is is significantly different between people with a low copy number compared to that with higher copy number. And notably, by the end of the 15 years follow-up time, you can see the red group with low copy number has reached like EGFR of 60, which is a clinically defined threshold for chronic kidney disease development in population level. So with all of these data, I mean, we are we are very interested to looking at that with cognitive function and cognitive impairment, because I think that mitochondrial function in lots of the animal study has been demonstrating to be associated with neurocognitive neurocognitive function and brain health. So I have to admit, I'm an early stage investigator and basically just introduced to this cognitive impairment. So there are still a lot to learn from me, and I'm sure that you guys are by far the expert in this particular field. So what we try to understand better is whether or not there is an association between mitochondrial DNA copy number and the cognitive function, right? So especially we noticed that there are some mixed evidence in the past showing that some studies show a pretty significant association between copy number and the cognitive impairment. Especially in the general population, we see that low copy number is predictive of cognitive impairment and disorder. But in some of the study, we also see mixed evidence that showing some studies suggest high copy number is associated with cognitive impairment. So which one it is? So what we do is we try to investigate the association between copy number and cognitive function by leveraging two independent longitudinal cohorts that I built my KO1 award upon, the ALIVE cohort and the MWCCS. So our hypothesis general is trying to, we hypothesize that higher copy number is positively correlated with better cognitive performance. So our idea is that because we see low copy number has been associated with so many poor outcome in the population setting, based on our previous study, we thought that, low copy number may be an indicator for either an aging related and lower biogenesis kind of marker. So we thought that it may predict a poor cognitive function. So our study setting, as I mentioned, we use the data and, the mitochondrial copy number I measure in both of the cohort for my case. So in both of the max cohort and the ALIVE cohort. So one of the my very motivated master student, Lydia Liu, she is taking a lead on this analysis. So for a little bit background knowledge for those people who might not be familiar with the max and ALIVE cohort. So max cohort is a prospective longitudinal cohort that recruited men who have sex with men. And then the ALIVE cohort is a community cohort, also long running, recruiting person who inject drug in Baltimore, Maryland. And both cohorts started in the 80s. So Max is just celebrated the 40 years anniversary. And the ALIVE cohort is about to turn 40 in a few years. The amazing cohort from amazing Lumit data generating from both of the cohort are really why we are able to conduct this study, trying to look at multiple aging related outcomes such as cognitive impairment. And both cohort invite all of their participants coming back for a semi-annual visit. And we do survey and collect their blood specimen and also trying to do physical examination as well as some of the special special assessments such as cognitive impairment. So in terms of the cognitive function measurement, there is a slight difference between the ALIVE and the max cohort. So in the max cohort, we measured the global, the speed information processing, motor function, attention, working memory, learning, recall, and executive function, including this here. And then there are a total of 6 domains. And then in the ALIVE cohort, we do measure also the global, the speed of information processing, the motor function, attention, and working memory. And then the learning and the learning and memory are sort of combined in one assessment, and then as well as the executive function. So as you can see, I'm leveraging the existing data and the work that has been built upon, especially Leah Rubin and Demani Picker from both of the cohort has done tremendous work to all of these neurocognitive impairment kind of neurocognitive function assessment. And the other method is about the mitochondrial copy number. So as I briefly mentioned earlier, basically mitochondrial copy number in the ALIVE and max cohort are starting from like DNA extraction. And then, in the ALIVE, that was DNA extracting from the Buffy code, and in the max cohort is DNA extracting from PBMC. And then we use qPCR to try to find the cycle threshold for the ND1, which is the mitochondrial probe, and then the cycle threshold for the RPPH one, which is a nuclear DNA probe. So basically, we just compute the delta cycle threshold between the ND1 and RPPH. And as you can see, this is like a rough measurement of the ratio between mitochondrial DNA copy number to the nuclear DNA copy number. And we did number of QC step. And we also recognize that despite the best QC we have done, the mitochondrial copy number are heavily influenced by the cell composition. So we adjust and standardize for a number of pretty critical measurement after we finish the initial assessment. So we adjust for plating effect, pipetting order, but more importantly, we adjust for the cell composition in both cohort. And we did several evaluation, what is the best or most important cell composition to adjust for. And we landing on adjusting for the platelet count and the white blood cell count. So after that, we did a statistical analysis and using the linear regression, basically trying to look at the association between the mitochondrial DNA copy number and domain-specific measurement. And then we also trying to look at the global cognitive function. So the models eventually are both as on top of the things that I mentioned earlier, also adjust for the HIV serous status, education level, smoking history, alcohol, and injection drug use. And here is the breakdown for the baseline characteristic for the participants. So on the left panel, that's the participant characteristic for the max cohort. As you can see, we included 306 participants from max. And then the alive cohort, we have 244 participants with both mitochondrial DNA copy number and cognitive impairment measurement. And the median age is slightly older among the max cohort than that in the alive cohort. As I mentioned earlier, max cohort recruit men who have sex with men, so everybody is male. And in alive, this is mostly a 50-50 split in the population level, but with more male than female in the alive. And the racial ethnicity difference, max is predominantly non-Hispanic white participant, and the alive cohort is predominantly Black participant. And in the max cohort, the education level is substantially higher than that in the alive cohort. So I think in the so this lower education level versus higher education level is even differently defined. And in the max cohort, the higher education is college degree and above. And in the alive cohort, the higher education is the the college and above here. So as you can see, like a very difference in terms of the demographic and socioeconomic status between the two cohorts. And in terms of the risk profile, I think alive is predominantly current smoker, and max cohort has much less of that. Alcohol use is somewhat comparable, and then alive is a lot. Although we recruited them as person who injected drug, about 50% of them are the current drug user among people with HIV. And 20% of that are people with HIV in their life. And injection drug use is incredibly rare in the max cohort. Okay, so next we are going to talk about the result comparing the two cohorts. So I'm going to go by this unadjusted model quickly. And then, so as you can see that mitochondrial copy number, so what annotated here, the beta effect is associating the unit of increase if you are high versus low copy number. So each one standard deviation increase in mitochondrial DNA copy number is associated with 3.74 of processing speed increase in the max cohort, and then 4.31 and 4.6 in learning and memory domain. And then it also associated with 3.59 in executive function, and then 3.14 for the global cognitive function in the max cohort. And on the other hand, for the alive cohort, every standard deviation increase in the mitochondrial copy number is associated with 0.13 of motor function, and then 0.12 with attention and working memory, and then 0.13 with executive function, and a little bit increase with the global cognitive function. I think the possibility for like the slight difference in terms of memory might be because of the cognitive function assessment are slightly different in the two cohort. And then also keep in mind that a live participant has so much influence from their consistent substance use, and that which also demonstrate to have major impact to cognitive function. So I'm not sure if that's somewhat impacting, the association we assess between the copy number and the cognitive function in the alive cohort. And then this is the adjusted model. And as you can see that most of the result seems like consistent compared to the slide before. And then here I'm going to also provide some visual sort of illustration for our data to show how the distribution looks like. The top panel here are the max cohort, and the lower panels are the alive cohort. And then as you can see, and I have mentioned that a higher mitochondrial DNA copy number is associated with a significantly better learning and recall. And then also the global function. And then in the alive cohort, it seems like predominantly associated with the attention, and the borderline associated with the motor, and then also the global cognitive function. Next, we're curious whether or not there is an interaction between HIV infection and the association between the copy number and cognitive function. So we did a stratified analysis in both of the cohort trying to assess the association. We didn't really seeing a major association between the copy number and the cognitive function across all domains. When we are looking at that in the people without HIV, we do see those associations reemerge. So it seems like those associations are primarily showing up in the people without HIV subset. And then I'm not entirely sure why. Maybe people in this seminar could shed lines on that. My hypothesis is that because those people with HIV has so many other factors that's influencing their cognitive impairment, maybe mitochondrial DNA copy number may or may not be showing up in this independent association. And also there could be other factor that's influencing mitochondrial DNA copy numbers, such as inflammation and other hypercoagulation and such could also playing a part and becoming an uncontrolled sort of unmeasured confounder. I think the other possibility is that mitochondrial copy number might be such an imprecise measurement in general. So we may need to really dig down deeper, trying to look at the cell type specific mitochondrial function or the overall mitochondrial health in terms of trying to determine the direct association between mitochondrial function and the cognitive function among people with HIV. Well, my conclusion, the key finding is we found a positive and independent association between the copy number and cognitive function in 2 independent cohorts. And then it's predominantly among people without HIV. And then there are several of next steps. We're certainly trying to understanding more the underlying mechanism of mitochondrial pathway and cognitive impairment among people with and without HIV. We are hoping to evaluate the copy number and other mitochondrial assessment and their relationship with the neuroimaging data, and trying to get a better picture what is the most significant kind of association or implication in terms of mitochondrial health with cognitive function and brain health. And then we do want to dig down deeper, trying to refine the analysis with people with HIV, trying to find out if there is any other measure confounder or potential factor that's influencing this association, and whether or not we are missing something in the people with HIV part, and trying to get a better sense, the role of mitochondrial function and copy number play in terms of the cognitive impairment. So I guess with that, I'm gonna to sort of bring it to my acknowledgement. So I think the NIAID supporting my KO1 award, trying to look at mitochondrial genetic and mitochondrial function and their role in the aging-related comorbidity among people with HIV. And then also I want to thank the ALIVE cohort and the MaxWise cohort supporting by NIH. And this longitudinal cohort infrastructure is really, what we were able to conduct this study and trying to look at multiple aging-related comorbidity outcome in the cohort setting. So I would like to also thank my collaborator and a live investigator listed here. And yeah, so I guess I will open up for any question and discussion.
Thanks, Jing. There's a lot of questions in the chat, so we can go from the top to the bottom. I think, Jules, you had the first question.
Yes, hi, thank you. Thank you for the talk. It was very interesting. My question basically is the effect of HIV and injection drug use. So in MAX, which is no drug use basically, or no injection drug use, did you see a reduced mitochondrial DNA amongst the men? And did you see a reduced mitochondrial DNA in the ALIVE cohort? And was there a difference in the amount of reduction between the 2 cohorts?
Yeah, that's a great question. So we did see that in the max cohort, people with HIV also have lower mitochondrial DNA copy number. We have a slightly smaller subset in the max cohort compared to that in the ALIVE cohort. In terms of like the drug use question, I also have data for that. I was debating whether or not I should show it, but 1 second. Yeah, so so we did also see a pretty major impact for substance use on mitochondrial DNA copy number. For example, this study conducting in ALIVE, we try to look at, people with current injection drug use. Those people showing in the orange bar are overall current injection drug use have a lower mitochondrial DNA copy number. And if you're current injecting cocaine, you also have a reduction, and then current injecting heroin also have a reduction in terms of their mitochondrial DNA copy number. Interestingly, the injecting of the substance have a much more striking effect on the mitochondrial DNA copy number then that is like inhale or consuming these in other mean. So I think that's an interesting observation as well.
So that's a very important point because, and then the question is current use versus prior use with a history of say 20 years ago. If you find current use is different than people who had a history but are no longer using it for many years, if they still have a reduced mitochondrial DNA.
Yeah, so I think in the alive cohort, it's very hard to answer this question because everybody did inject drug to be as part of the recruitment criteria. So what we show is like continual injections does seems like having a continual impact. So I don't know, like, so even among those people, or like everybody here in this study is exposed to injection drug use decades ago, and then, you know, the current injections still have a significant reduction in the copy number. So I thought that's kind of interesting finding.
Yeah, no, the question remaining is if you used it 20 years ago and you stopped, will you still have a residual effect, a legacy effect?
Unfortunately, we don't have the answer for that, at least not in the alive. Maybe we can investigate that further in the max cohort with, you know, a little bit like people with much less exposure to injection drug use. And then maybe we can try to see if we can differentiate current injection drug use, past injection drug use, and never use. Maybe that's a better way to answer that question. Yeah, but currently I don't have that data.
I think it does have a legacy effect, and I just want to end by
Could
saying
be.
if you
Yeah,
use
I just
both,
don't know.
if you
Yeah,
use both together, heroin and cocaine, it's probably even a worse effect.
it could be. And keep in mind that the figure I'm showing here is actually already like an independent effect of both. So it's like the independent effect of each of the drug use.
Thanks, Jules. Shan, do you want to unmute yourself and ask your question?
I think she already addressed my question after I posted the question in other slide. I think that's not the whole mitochondrial DNA sequence, but just the QPC.
Yeah, it's not the sequence. We were planning to do that work. So good point. You're reading my mind. So yeah. So this is like my KO1, which has like the very low research budget, and that's all I can do. So
It's a good start. Yeah.
thank you.
Hilwa, if I'm sorry if I'm mispronouncing. You want to unmute?
Oh, hello. Actually,
Hello.
I'm quite new on this community. I'm from Morgan State University, and recently I was funded with Dr. Amanda Brown at Hopkins. So I'm still on that mentality of beginner with best of luck as a beginner, actually. But I'm very happy to further well nurtured and well cultured on this fascinating cooperation and collaboration. So I'm actually not an expert in mitochondrial DNA related study, but I recall mitochondrial DNA is only inherited from maternal, motherly genetics. So accordingly, one beginner's question in common sense is on that in and out in between cytosolic RNA and possibly retroviral kind of RNA velocity vector fields from RNA to DNA and DNA to RNA. There any sexual dimorphism, so sex dependent kind of genotype to phenotype kind of mapping. That is one question, intriguing question.
Yeah. So
of
I
mine.
think, yeah, I think we did you know, we didn't really do a sex specific stratify analysis here. So but we do see in general, women has lower mitochondrial DNA copy number. So there is a previous study that shows that there is sex difference in terms of mitochondrial function. So I do think that that's probably something that's really worthwhile to investigate. So
Thank
That's
you.
Thank you.
Scott. Dr. Latender.
I am driving. Can you hear me?
Yeah.
So very nice presentation. I had a couple of questions. We had done some work in this area with Todd Hulgan and David
Yeah.
Samuels at Vanderbilt.
Yes.
And they deserve all the credit. They did some beautiful work. But one thing that I recall learning in the process was wishing that we had sorted or selected. And you
I'm
know,
sorry,
that's
yeah, I missed that. I missed what you were just saying. You wish you were sorted. Did you mean like sorted the cell?
yeah, cellular subsets. Either sorry,
Right.
I went through a bad cell phone area here. So either sorting or selecting cell subsets like monocytes or T cells, CD4 positive T cells, because there is both differences in the energy demands of those cells, but also differences in the pathogenesis of how HIV affects the brain.
Absolutely. Yeah, Scott, I'm very aware of the work that you mentioned. So, so I, one of my slide talking about like mixed evidence of copy number and, cognitive function is, like I was thinking about the paper that Todd's group have done in the past. So yeah, I think this is a great point. And as I mentioned that mitochondrial DNA copy number and have such a high heterogeneity across cell type, in blood. So I think if we have the resource to sort it, it's absolutely going to add so much value to this measurement. So what we don't have at the time is that, we don't really necessarily have the resource to sort all of the cell, but then what we did is we leveraged the cohort infrastructure and we try to use the CBC data that's already exists in both Alive and Max. And I know that this did not get the cleanest cell type specific mitochondrial copy number or proxy of mitochondrial function, but at least it's better than the crude assessment because it has a huge effect from the platelet count, and we know that people with HIV has differential platelet count, and then also HIV impact T cells, so the cell composition is very different from people without HIV. So yeah, and also I currently have an NIA supplement trying to look at cell type specific mitochondrial function. So in that work, we didn't sort them, but we were able to get down to like the single cell level, the cell type specific mitochondrial function, and we are working on the CROI abstract on that. So hopefully can share more of those with people in the near future.
One other suggestion in the charter, at the Baltimore site of the charter cohort, they have the highest proportion of people with hepatitis C co-infection, and many communities like yours and ours now have harm reduction programs that reduce hepatitis C, but in many of the stored specimens, people may still have had it, and that's another highly immune stimulating
Absolutely.
viral infection that could also affect mitochondrial DNA. So
Yeah.
sorry, I don't recall if you controlled for hepatitis C or not.
I did. So yeah, I did control for hepatitis C. However, I did see striking effect for hepatitis C with the mitochondrial copy number and function as well. So this is definitely if not the most, at least the similar impact as HIV as a chronic infection impacting the overall mitochondrial health. So very great point, and thanks for the pointing to the resource.
Okay, thank you.
Thanks, Scott. Mira, do you want to unmute yourself?
Yeah, sure. That was a really great talk. I feel like I learned a lot. I just had a quick question about like the cognitive testing measures. Because I know that the Max cohort reported the cognitive testing scores as T scores, and the Alive cohort reported them as Z scores.
Yeah.
And I know, like, for T scores, sometimes the normative distribution is different. Like, say, if you were like a 30-year-old woman, they would compare you to a distribution of like only other 30-year-old women instead of just the whole population. So I just wanted to ask if you know whether the Z scores in the Alive cohort were also like adjusted that way.
Yeah, so it did adjust it for, although I do know that there is a difference between the two. I mean, I'm going to look at Leah and all of these data science, expert here, and maybe we will need to maybe it is necessary eventually to try to harmonize the two cohort, at least using the two, the comparable Z score to do it or consistent T score to do it. And I also wonder whether or not the effect size difference, as you can probably see earlier from this slide, the effect size seems like quite different. And I wonder if that's also related to the different T score and Z score we are using, and maybe leading to this kind of effect size difference here.
Yeah, maybe.
Yeah, I was gonna say, Jing, like I know way more about the Max and the Ys of their demographic norming. I think it was like Ulna Celes. There were some folks that were involved with their more in-depth cognitive assessments before I arrived here at Hopkins. And then we do have, we have computerized assessments that we have been creating demographic norms, but that's not here. So I don't know the exact, I'd have to go back. But yeah, you bring up an important point.
Yeah.
Thank you.
Yeah. Rob Paul.
Hi there. That was a great presentation. Thank you for that. I had a couple of questions, but I'll limit to just one. I kind of following up on the cognitive measures. It seemed like there were some pretty extreme scores in the data sets. T scores even at 0, which is unusual. And then there was a fair amount of aggregation of scores kind of in the center of the scatter plots. I just wonder if you had a chance to look at to what degree the extreme scores might have influenced the overall results.
Yeah, I think that's a very, very good observation. We have not done, removing the outlier and the extreme score, but I think it's probably worthwhile to investigate. Noted that the one that we show significant association, like learning and record, they don't really have the extreme measure in those domain. I think the one that like the motor for the Max and maybe the motor in the Alive. So both are like the motor does have some extreme value there. So maybe we should look at that specifically.
Yeah, and just that scatter might also suggest that there's some nested subgroups that are layered in there that would be you might be able to flesh out with some type of clustering analysis.
Okay.
You are spot on, Rob Paul, and we've actually put in the request to Greg to try to get that data to harmonize it with the other data.
That's why your brain and my brain are part of the same brain, Leah.
No, so yeah, so that is the hope. Greg is, he's constantly running around. So hopefully, sometime in the near future, we'll be able to do that and then kind of do that harmonization and see what happens.
Cool.
But yeah. Hemo.
We've been chatting on on the side about
Oh,
this,
yeah.
basically just correlating these absolute numbers of mitochondrial copies with functional assessments like seahorse. But it sounds like there's people already working on that. You guys might be collaborating already.
Yeah, I gave as part of my glucocorticoid study where we were giving hydrocortisone, Gretchen Nye, and was looking at glucocorticoid receptor function, and her MD PhD student did the seahorse assay on those. So she is writing up the abstract right now to go into CROI, and not only looked at it with correlation, but looked at it in the context of, you know, a phase 0 clinical trial. So I'll let her share those findings. Hopefully I can get her to come on in the spring. But yeah, and Jing, you may have some more thoughts on that.
Well, I do have a quick question for the seahorse assay that you guys run. Did you use the frozen variable PPMC, or what do you use to, or the
I
fresh?
mean, we
or
send frozen PBMCs to
Oh,
Gretchen. So yeah, but you can talk to Gretchen and Hannah. Hannah comes oftentimes to our Friday meetings, but she can tell you all that you want to know about what I mean, I
That's
don't
cool.
run
Yeah,
that.
I'll try to join when I'm teaching this term with the class
Yeah.
that just overlap with that meeting time, but I'll try to see if I can join that later. Yeah, I think seahorse, some of the previous work that you know, working with my collaborator, we all maybe we just didn't nail the technique. So I'm curious to learn more about that, because there are a fair bit of inconsistency for the seahorse assay. So I wasn't sure if it is yeah, relevant to, yeah, maybe it's the technical aspect of that, of running that assay.
Any other questions? That's everything that's in the chat. I love this. We've got a lot of, you know, back and forth. So it's been great. Any other last minute questions that anybody wants to unmute themselves?
I have
Jules.
one more question. This is Jules. Hi. So, what's the chance? I know you probably don't have data on this, or probably nobody does, but my thinking is that for injection drug users, people with a history of that or current use even, what's the chance that that factor makes people with HIV more vulnerable? So the combination of the two makes HIV still a factor, but a worse factor. So it's a combinatorial effect.
Yeah, I think that's a very good point, Jewel. I think there are lots of evidence that cocaine use and heroin use or other opioid use does you know, thus associated with HIV viral control, and then early on there is a, you know, like aggregated effect of the substance use and HIV. And in the you know, current era of ART, although HIV viral load is well suppressed with the ART, substance use, one is associated with suboptimal taking of antiretroviral therapy. Therefore, you know, a lot of people, like, especially like when you're looking at the alive cohort, people are having this suboptimal taking of ART due to their constant substance use. So that is one factor may lead to a poor HIV related outcome. And additionally, the cocaine use and opioid use does show some evidence to increase chronic inflammation, and that compound with the chronic inflammation related to this low-grade viremia from the HIV, even under effective ART treatment, I think the compound impact for aging-related outcome is huge. And then I'm not surprised at all that's leading to like accelerating aging and such among people who consistently using substance. So yeah, definitely the compound effect of the two is pretty lethal. Yeah.
And I would just add that, you know, we have several global neuro researchers from outside the US who claim that HIV does not affect cognitive impairment, generally speaking. And I don't think they're considering these kinds of factors at all. So I think there's a difference in the populations between what they're talking about and what we see.
Yeah, I agree 100% with you. I think even well-controlled HIV with effective antiretroviral therapy, there is evidence showing the viral reservoir of HIV and then the constant low-grade inflammation caused by these, low-grade viremia could be translate to neurocognitive inflammation, aging, and then neurocognitive impairment. So at least I think there is a clear link there. How well we can tease out in the population level just takes a lot of work from us, so we're constantly working on that.
And Jules, Rob and I think, was it last year, Rob, or with the data from Uganda, it's now, being submitted. It's submitted for review this week. But we clearly showed difficulties in people with HIV with higher levels of impairment, and very similar groups and way less confounding factors. So I do believe it's present. Rob may have more to say because he works all over the world. And Scott as well, I know other folks do.
Yeah.
Rob Paul, do you want to say anything or?
Hi, this is Pim. I say something. What you need to
Hi, Pim.
is cognitive reserve, because that
Yes.
may be something that's specifically different in populations that you look at. And I'm not sure how much people take that into consideration. Maybe that's actually the difference between some of the European cohorts and the cohorts in the US.
Yeah. Thanks, Pim. Any final comments? Jing, thank you so much for presenting here today. Caroline. Oh, go ahead, Jing.
Oh, no, I just want to thank everybody for these great comments. I've already taken tons of notes on that.
No, it's amazing. And then Caroline, do you want to present who our speaker is for next week?
Sure. It's Kalen Peterson, and he's talking about biological aging, brain imaging, and cognitive impairment in persons with HIV.
Awesome. So can't wait to hear Kalen speak next week. He works with Boansis out at WashU of St. Louis. And so have a great rest of your Wednesday, and we'll see you back here, same place next week. Bye, guys.